metabolism. And I like to start off with asking the question, what is fermentation? Because most people talk about it, but I don't think they know biochemically exactly what it means. So I've broken it down into 2 bullet points. First of all, it's the breakdown of organic compounds, particularly sugars or carbohydrates, to generate ATP by a process called substrate level phosphorylation. So substrate level phosphorylation occurs when the substrate itself has a phosphate group on it, and an enzyme will transfer that phosphate group directly to ADP to make ATP. In fermentation, though, the terminal electron acceptor needed to regenerate oxidized NAD, or NAD+, is derived from the substrate, so there's not an external electron acceptor. And so I'll show you that when I get into the actual details of the pathway. So both of these need to be met in order for it to be considered fermentation. So a quick overview. The net energy yield is pictured over here, and the flow of carbon is pictured in the middle. And so essentially what you have here is the breakdown of a sugar, a 6-carbon sugar, into 2 3-carbon pyruvates. And then these pyruvates are used to produce the various end products characteristic of the type of fermentation. So they can be acids and alcohols and certainly different kinds of gases. If the organism is capable of oxidative respiration, like you and I do, for example, or an aerobic bacterium, or a yeast when it's undergoing aerobic respiration, the pyruvates can be shunted to the Krebs cycle, and then electron carriers generated from Krebs can be used to generate additional energy through the electron transport system. So over here you see that there's basically an output of approximately 2 ATP molecules per sugar, and you invest oxidized NAD or NAD plus and produce reduced NADH, and the buildup of this reducing power here, because these are fermentative organisms, has to be depleted. And so those NADHs come over here and dump their electrons into the terminal electron acceptor, some carbon skeleton in the pathway, to produce the desired end products. Some of the types of fermentation, and I've highlighted the one that's important for the class, most important for the class, alcoholic fermentation. But butyric and butanediol and mixed acid and propionic and lactic are other varieties as well. And so these cartoons sort of give you a walkthrough on the different kinds. So here's a central sugar, and then an organism undergoing butyric, like Clostridium perfringens, would produce these types of end products. Solvents like butyric acid, very bitter aroma, and alcohols and acetone, another solvent, and CO2. And so the type strain here, Clostridium perfringens, also causes gangrene. So if this guy gets into a deep puncture wound, it can grow and produce toxins that can destroy the tissue. But they also ferment muscle glycogen to produce all these compounds and a ton of gas. And so the reason why this individual, and I don't know if you can see it here, is stitched up here, is the ankle had swollen so much that the tissue started to burst, and they had to cut it open and remove as much of the gas and dead tissue as possible. Down here you see additional types of fermentation. One is butanediol. So a version of this is important in wine microbiology, and I'll show you that in a bit. But here you're seeing it used to conduct the VP, or Voges-Proskauer test, in microbiology labs. And so you can feed the microbe glucose and then ask the question, does it undergo butanediol or mixed acid fermentation? And that's diagnostic for different kinds of microbes. So a butanediol fermenter will produce, as the name suggests, butanediol and CO2. Don't worry about what that is. And then a mixed acid fermenter will produce a variety of acids. And again, you can use basically a pH test to tell the difference between these types of organisms. But again, I'm not going to ask you questions about MR and VP testing. I just wanted to put it in there to give you some context. More important to food microbiology and wine microbiology is this slide. And so here you see a central sugar, microbes undergoing lactic fermentation and producing lactic acid, ethanol, acetate, and CO2, which can produce the variety of cheeses we're used to seeing, or yogurt, or a variety of things. And so down here then is our alcoholic fermentation, where the desired end product is ethyl alcohol and CO2. And so again, you can see the CO2 manifesting as a nice creamy head on this beer, or the zing in a champagne. The type organism that we use is typically Saccharomyces cerevisiae. Propionic fermentation is the result of fermentation of end products of lactic fermentation, typically. So the microbes that are lactic fermenters will produce these compounds, and then these compounds can be used by propionic fermenters to make propionic acid, acetic acid, and CO2. So propionic acid is the primary flavoring agent in Swiss cheese, and the CO2 is responsible for the eyes that form in Swiss cheese. And the microbe that we use typically is a Propionibacterium species. This is just a little bit about the biochemistry and microbiology of lactic acid fermentation, some of the general principles. So the types of lactic fermentation include homolactic, where lactic acid's virtually the sole fermentation product. And we typically use homolactic fermenters in the first stage of cheese manufacture. So when I'm making cheddar, for example, I'll use a Lactococcus species that is a homofermenter. Heterolactic fermentation is a bit more complex, and here microbes produce a variety of end products such as lactic acid, ethanol, acetate, and CO2. And so something more complex than cheese, like sauerkraut or kimchi, we can use heterolactics to perform those reactions. So here's a quick overview of the biochemistry of homolactic fermentation, very stripped down version. So here we start with a 6-carbon glucose. We have an input of 2 ATP to make a bisphosphate that you see here, so fructose 1,6 bisphosphate. We have this central enzyme, the fructose 1,6 bisphosphate aldolase, which cuts this guy into 2 3-carbon molecules. And these individual G3Ps then can be used to generate ATPs, so by substrate level phosphorylation. So these phosphate groups can be tagged onto ADP to make ATP. But because this is fermentation, we like I said, we generate a fair amount of NAD plus that and as a result of that, we have to pick up some of those electrons and stick them onto NAD plus to make NADH. And then, of course, we can't have an over excess of reductant, so the electrons picked up by NADH have to be dropped off to one of the intermediate substrates. So pyruvate, in this example, picks up these electrons given to it by lactate dehydrogenase. So again, lactate dehydrogenase will pull electrons off of NADH, stick them onto pyruvate, and give us lactate, which is our major flavoring agent in cheese production, and it also helps form the curds. The acidity helps form the curds. Heterolactic fermentation, or the phosphoketolase pathway, is a little bit more sophisticated. In this particular case, you've got a 6-carbon sugar, and you have an input of 1 ATP to make this xylulose 5-phosphate. Again, you have a step here where electrons are being pulled off of the reduced substrate and passed to NAD plus to make reducing power. And of course, the reducing power, as it builds up, has to be spent. And so that happens down here. So the 5-carbon phosphate is cleaved by the phosphoketolase, well, after it's phosphorylated, cleaved by phosphoketolase. And so you end up with G3P phosphate and acetyl phosphate. So this is basically just like homolactic fermentation, as I showed you before. And so the G3P phosphate can be used to generate more energy, and the resulting pyruvate can pick up electrons from NADH so that we can regenerate NAD+. Acetyl phosphate, as you see here, can proceed in this direction to produce ethanol, and it's another way to burn up some excess reducing power that we've generated in different places. And so the electrons here are transferred to the acetyl phosphate, and we ultimately make ethanol. and that regenerates the NAD+, which can then go over here and serve as an electron acceptor at different stages. And so in this particular case, the other option that microbes have that are heterolactic is if there's a tiny bit of oxygen around, this reaction, the production of ethanol, is de-emphasized, and this reaction actually is emphasized. And the good thing about this reaction to produce acetic acid is, well, it's a flavoring agent for one, but the microbes can also make ATP from this. So again, you can get a substrate level phosphorylation event where this phosphate is used to make ATP. And that's only in the presence of a little bit of oxygen. And the reason for that is oxygen can make this reaction happen spontaneously, and so that we don't have to worry about dumping these electrons off to make ethanol. And so again, the organism can favor this reaction and generate 2 ATP. Normally, heterolactic fermentation generates 1 ATP. I'm sorry, yeah, 1 ATP from the breakdown of glucose. As you can see here, we've made 2 at this step, but we had an input of 1 early on. So 2 minus 1 gives us 1 ATP. If the microbe can do this, then it can make 2 ATP total from each glucose. So the way we visualize this in the lab is through these phenol red or BCP lactose broths. And so here's an uninoculated broth. It has a purple pH indicator in it with a Durham tube, and you can see that. And so this organism is a heterolactic fermenter, and so you can see the bubble formed in here as a result of the fermentation of lactose in this tube, acidification, and then the purple pH indicator turns yellow. Over here, it's a homolactic fermenter, no bubble in the Durham tube, and it's because it's just producing lactic acid. This is an overview of some selected genera of lactic acid bacteria that are used in food, in some cases wine production, and some are just wine spoilage microbes. But the genus name is over here, the morphology of the organism pictured here, and then the type of fermentation, and the lactate isomer as well. This isn't so important, but I definitely want you to focus on this stuff here. So the first organism, Lactococcus, typically it's used in, for example, cheddar production, cheese production, and the morphology, as the name suggests, are cocci, and they, sometimes in chains. They're homofermenters, homolactic fermenters. Here's Leuconostoc and Enococcus. Enococcus, hopefully that name rings a bell to you. It's used in malolactic conversion in wine production, but these guys look a lot like Lactococcus, for example, but they're heterofermenters. And the Pediococcus, they're often spoilage microbes in wine. I'll talk about that in a different lecture. But again, the coccus suffix in their genus name suggests their morphology, and they're also homofermenters. And then the Lactobacilli, so these are chains of rod-shaped organisms that can be homo or heterolactics. And then Streptococcus are pretty good fermenters that we use in yogurt production. Well, certain species of Streptococcus we use in yogurt production, and they're homofermenters as well. So here's a quick overview of ethanolic fermentation in yeasts. And so we begin with the 6-carbon glucose. It's a basic glycolysis process where we have 2 3-carbon glucoses that are cleaved ultimately to form 2 3-carbon pyruvates. We use 2 ATP to make 4, so we net 2 ATP overall. There's a decarboxylation step here where the 3-carbon pyruvate is decarboxylated to a 2-carbon acetaldehyde, and so there's acetaldehyde. Acetaldehyde then serves as the terminal electron acceptor to pick up electrons from NADH to form ethanol. And then the oxidized NAD plus gets cycled back to pick up more electrons, and we at least can complete this cycle so we don't build up reductant, for example. The desired end product is ethanol,